Good afternoon to Honorable Dean Sri Tanmay Singh Sir, Administrative Officer and the HOD of CS2 Department Sri Rana Chakrabarti Sir, the HOD of Mining Engineering Department Dr. Shankar Sarkal Sir, HOD in charge of Mechanical Engineering Department Sri Abhishek Mitra Sir, and Sri Shankar Sarkar, Lecturer, CS2 Department, and one and all present on board. At AIEMP, Asansol Institute of Engineering and Management Polytechnic, we believe in impacting the minds of our students with trend-setting ideas. And henceforth, our worthy management have decided to organize Impact Lecture. Today is the second day of Impact Lecture. And at this moment, we have been joined by our esteemed keynote speaker, gem of a scholar and a researcher, Dr. Gorachand Dutta sir from Kharagpur IIT. Sir, we have been looking forward to listen to you, and we hope this session will be a memorable one. But prior to that, let us celebrate the moment with lighting of the lamp ceremony. We have our Honorable Dean Sir, Administrative Officer, and the HODs and Shankar Sarkar Sir to light the lamp and make an auspicious beginning of this program. Sir, please. Now, let us all wish a warm afternoon and accord a heartfelt welcome to our esteemed keynote speaker of Impact Lecture, Dr. Gorachand Dutta sir, Assistant Professor, School of Medical Science and Technology, IIT Kharagpur. Sir, we are truly honored to have you as the keynote speaker of Impact Lecture, and we are sure that your words, your ideas will empower us all with trend-setting ideas. But prior to that, let us hear our honored Dean Sri Tanmay Singh sir, who with a speech will make the beginning of this program. Sir, please.
Good afternoon, respected Director Sir, Principal Ma'am, Administrative Officer Rana Sir, all HOD and HOD in charge, Sako Sir, and Honorable Dr. Gorachand, Dr. Assistant Professor of IIT Kharagpur. Today, we all gathered here to innovate the Impact Lecture series, as it is honored for us that it has been funded by Institute Innovation Council and AICTE. We are the first polytechnic to have this grant. What is Impact Lecture? Impact Lecture will enable good knowledge towards the emerging trend in student. As Sir will deliver on the topic Nano Biotechnology Enabled Biomedical Sensor Device for point of care disease diagnosis. We are very much eager to hear the sir as a speaker. As before, I have also heard these types of webinar from honorable speaker. Thank you, sir. We are very much eager to have your speech. Thank you.
Thank you, sir, for bringing out the significance of Impact Lecture 2022. Now, may I request our administrative officer and the HOD of CST department, Sri Rana Chakrabarti sir, to deliver a few words for all of us.
Thank you, Devasith sir. A very good afternoon to our respected Director sir, Honorable Principal Madam, our keynotes, Speaker Mr. Gorachand Dutta sir, Dean of Academy Mr. Tanmay sir, all HOD in charge, faculty, staff member, students and all viewers. This is a great time that we have with us Dr. Gorachand Dutta sir, Asst.Professor IIT Kharagpur. This is our fourth sessions of this Impact Lecture series, and we are very much honored to have sir with us today. Hope it will be a very great sessions. Students, viewers, all of we will be. We are eagerly waiting for this lecture series, and hope it will be a great time for us. Thank you.
Thank you, Rana Chakrabarti sir, for your inspiring words. Now, may I request Dr. Shanku Shaurkel, the HOD of Mining Engineering Department, to deliver his thoughts for us.
Thank you, Devasith sir. Thank you, Devasith sir. Very good afternoon to all. Respected Director sir, Principal Madam, our honorable guest speaker, Dr. Gorachand Dutta sir, Dean, Mr. Tanmay, all HODs, professors, and dear students. I welcome you all to this Impact Lecture hosted by Asansol Institute of Engineering and Management under IIC, AICT funded Impact Lecture Series. We are really honored for our guest lecture's presence, Dr. Gorachand sir, for sparing your valuable time to deliver the lecture on emerging trends in science and technology. I hope your lectures will be hugely benefited to the students and for the viewers later on. Once again, I welcome you, sir, for this Impact Lecture, and thanks for being with us. Thank you.
Thank you, Dr. Shanku Shaurkel sir, for your inspiring words. Now, may I request Sri Abhishek Mitra, HOD in charge of Mechanical Engineering Department, to grace the occasion with his words.
Thank you, Devasith sir. Good afternoon to Director sir, Principal ma'am, Dean sir, Administrative Officer, all HOD and HOD in charges, all the students and those who have joined this session. I would like to cordially invite Dr. Gorachand Dutta, Assistant Professor, IIT Kharagpur, to give his valuable lecture on his second day of Impact Lecture, funded by IIC and AICT in our college, that is Asansol Institute of Engineering and Management Polytechnic. The topic, that is nano-biotechnology enabled biomedical sensor devices for point of care disease diagnosis, would be a valuable lecture for the students, those who have been debarred from the knowledge during the COVID. Hope this session will have a valuable impact on our students and benefit our students in their future. Thank you. Thank you, sir.
Thank you, Sri Abhishek Mitra sir, for your encouraging words. Now the opportune moment has come. Now the opportune moment has come, and we all have been eagerly waiting to be a part of this session and listen to our eminent speaker, Dr. Gorachand Dutta sir, Assistant Professor, School of Medical Science and Technology, SMST, IIT Kharagpur, India. Dr. Gorachand Dutta sir did his BSc in Chemistry honors from Vidyasagar University, and then his MSc in Chemistry from IIT Guwahati, and then he did his PhD from Busan University, South Korea. As an outstanding scholar, he has left his mark in the domain of academics, and we are very sure that his words will uplift our young minds and encourage them to do something worthwhile in future. Sir, we are proud to have you with us this afternoon, and we are very eager to listen to your priceless words. Sir, the microphone is all yours now.
Okay, so thank you very much. So can you see my screen?
Your screen.
My
Yes.
slide screen, is it visible? And I am changing my screen.
Yes,
Can you
sir.
see?
Yes, sir, we can see it. It is absolutely visible.
Okay, I'm changing my slides also, you can see, right?
Yes, sir.
Very good. Okay. Okay, thank you very much for your invitations and let's-
May I interrupt with your kind permission? Yes, sir. So may I say something? Sir, your camera actually probably is not in proper condition. Yes, sir.
Is it okay now?
Yes, sir, it's better now. It's much better now.
Okay. Okay, fine.
Thank you, sir.
So thank you very much for your introductions and let's start the topic. So today I'm going to introduce some new technology for different kind of disease diagnosis, where we use different nanobiotechnology that can be used for sensor development. And during the pandemic time, like COVID time, also it's urgently needed some sensor device that can detect the COVID, especially in the near patient, or maybe by yourself you can check and you can take the decisions, something like this. So this kind of technology I will show you that we are developing at IIT Kharagpur also, and so something that I and my group already developed during the different times, and also I collaborated with different institutes all over the world. So this is the basic outlines of today's presentations. So first things is just kind of introductions I will mention, then different kind of disease. Also, then we'll discuss about the commercialization process of this kind of sensor devices, because final our goal is these innovations should be for the mankind, for the society should get the benefit from this kind of innovations. So there is so many future prospects also in this kind of research area that also my students you can learn. Also you can focus also in future. So this is very common sentence, like health is wealth, right? So why this healthy choice? Because if you are not healthy, you cannot enjoy the environment. You cannot enjoy our beautiful world. So we should, I mean, take care of our health. But sometimes it is very difficult to maintain our health. That you understand during the COVID pandemic time. Although you are very much careful, but you don't know something it can affect your health also. That's why we should take the proper decisions, proper precautions. So who can help you with this? Some new technology, kind of device that you can detect your disease in the early stage, and then you can take the precautions. You can also not only yourself, like your family member, you can take care of your family members. Just think about the COVID. If you are affected, you can spread this on for your family members also, right? So this kind of technology that is called the point of care. Point of care means it is just bench to bedside. Means the bench means that is where we are developing inside the lab that can be useful for the near patient. Even the end user like you can use in your home and take the necessary decisions. See, why we are interested in this research area, you know, not only COVID that you are affected, there is so many other diseases also like HIV, malaria, tuberculosis. This is kind of the old state problem uh, all over the world. Also cardiovascular disease like heart disease. This is kind of the number one cause of the death. Right, you know this one. Like in India also, you see the different regions. You can okay, I can see the laser pointer. You can see. So in all over the India is a different part. You can see that different region they are actually affected of the heart disease. This is the number one cause nowadays. Also, more than 50% were people suffering different chronic disease like diabetes. You know, right? So for this, we will take the proper. I can say that some technology so that we can detect the disease at the early stage and we can take the proper actions. But what the current solutions nowadays? You know, so nowadays generally you have to give your sample, then you send the sample to laboratory, they will process it with very high end and the instruments, and then they will send you the data, and then you will get the information you have this problem. But this is fine for some like developed countries or who are very rich. Sometimes very expensive for diagnosis also. But think about the common people to say rural areas or developing countries. Sometimes we are not taking care of much our health because we don't have the access of this kind of setting. Like we don't have this kind of high-end instruments facility near the hospitals in the rural area. Then how a very common people in the rural area they can think about the early stage disease, right? Sometimes the cancer at the end stage and they came to know they have cancer. Then what's the choice? Then we have to wait for at the end day of his life. This is the, I mean, we have just we are thinking. Like if we can develop a very simple device like the glucometer, you know, it's kind of cheap. Like I will show you the different very simple devices. Just you will drop a single drop of the blood samples or any kind of saliva samples or urine samples, and you will know the problems, including cancers, other diseases like HIV, malaria, tuberculosis. There is so many other infectious diseases, right? So if you know that you have this infectious disease, then you can take the precautions, not only for others, also for your family members, so that it will not spread, right? This is actually main goal to develop a point of care device. See, this is the one solution that I'm going to say is the biosensors. Maybe many of you don't know what is the biosensors. So biosensors is a kind of the analytical device that can detect very specific disease with the help of some biological recognition element. So these recognition elements are very much specific for a disease. So suppose you want to detect the malaria. So this biological element specifically detect the malaria and let you know that you have this disease or not. or COVID, for example, SARS-CoV-2 virus. So we can use some very specific biology recognition element that can detect specifically COVID and give you the information. So what the kind of biorecognition example? The example is the antibodies. They are very much specific. Example, the peptide, aptamer, different kind of enzymes also we can use for different disease diagnosis. Also, we can use the DNA RNA also. On the left hand side, you can see this is a very simple a schematic for biosensors. So before that, I will show you the different kind of biosensor technique. There is many biosensors available, like electrochemical biosensors, calorimetric biosensor, mass biosensor, thermometric biosensor. What is the electrochemical? That is the schematic here. Means schematic here is showing that on a solid surface, that is called the transducer, where we remove the specific biological recognition element that just to describe like antibodies. Then we will draw the samples. So if your samples contain the specific disease marker, that will bind on the surface and will apply some potential, some electrical potential will apply. Then some electron will flow from these transducers, means you will transfer your reactions, when some reactions happen on the transducer surface, it convert this into a biological signal, and from this signal to an electrical signal. So because of the electron transferring through the transducer, that signal you will get in form of current, in form of charge, and you can measure. So if the concentrations of your target, like for example, like the SARS-CoV-2, you are measuring. If concentration increase, you will get accordingly similar proportional current or charge that you can measure. So this is a very simple concept. Actually, as I mentioned here, there are different kind of sensors available. From there, actually, we are interested in just I described the electrochemical sensor. We are very much interested on this. There is some points why we are interested in electrochemical, not the others. Not the others with colorimetric change. Colorimetric also there is some color change. Means if there is no color, then you can say there is no disease, no virus. If there is color change, then we can say there is a virus. Mass biosensor, there is a mass change. Thermometric biosensor, there is a temperature change. But among these, the biosensors, actually, electrochemical biosensor is a very, very important a biosensor, and we are interested. Why? Because it's a very simple technique. And not only simple, it is very, very sensitive. As I mentioned, the colorimetry, like there is no color, mean I can say no virus. But sometimes there may be some virus present, but very, very low number. That time you cannot see the colors change. But electrical current, electrical charge, that we can measure. So that's why if you, electrochemical biosensors are very, very sensitive at the very early stage, like cancer. So if you go for the colorimetric change, maybe sometimes it is not possible at a very early stage. At that maybe middle or end stage, that time it's difficult for the go for the treatment, because there is sometimes you can say cancer, there is no answer. So if there is a very late stage, we cannot save the life. That's why we have to detect the early stage. Like at the same time, like the COVID, if we can detect the early stage at the very beginning, then we can stop it spreading. We can stop spreading. Electrochemical biosensor can help because it is very, very sensitive technique. Sometimes we are saying this is ultra sensitive technique. Right? Very small amount, if it is present in the biological fluid, we can measure. Also, easily we can miniaturize. So why it is important miniaturization? Because very bulky instrument, we cannot bring, we cannot easily, you cannot keep in your home, right? Or you cannot check, like even you can keep a sensor in your pocket. But for the if it is very bulky, you cannot keep. Right? Very small device you can make, and you can keep in your pocket, and you can use this one in some critical states for like that for the diabetic persons, right? Maybe sometimes their glucose level can increase very high. So before meal they can taste it. They can keep in the pocket and they taste it and take the how much sugar they should consume or not, right? That they can take the decision by themselves. No need to depend on the doctors also. And it's affordable. So it is very, very important, especially developing countries and rural areas. So it should be affordable, not that much expensive. It is very expensive, then that is our main goal, that society should be benefited. If our people, if they not buy it, then what is the use of this kind of development? So we are interested in this. Nowadays, one thing I think you have to think, many people now almost, I think they have their smartphone, right? Because of the technology growth and development. So we can integrate some biosensor, some sensor with your smartphone, so that by using an app, you can measure the amount of the biomarker. What is the biomarker? That is your disease analyte that we can detect. That analyte means the disease marker that you can detect. So, for example, for the COVID case, SARS-CoV-2, your analyte that you are going to detect. So, no need any kind of pre-processing, sample processing. No need any kind of specialized persons who can help you. You can do by yourself. Also, the multiplex, that also we can develop by using the electrochemical sensor. Multiplex means not only the one analyte, maybe more than one. Maybe it's at the same time, see, sometimes this time malaria or dengue, both can be affected, right? So in this case, we can develop a sensor that can guide you if this kind of both present or only single virus present. That also we can get. So this is the things, like we can use some, we can develop some printable. Also we can use some sensor that can print on the clothes, and that can detect different kind of disease marker from the not only blood sample, like from the sweat samples. And the label you can detect by using your smartphone, also by using the IoT, the Internet of Things. Nowadays it's very, very developed technology now we have, and if you have any like critical conditions, like before the heart attack, something like this, we can send the information also to your smartphone. Also, you can sometime maybe you don't have any condition to take the action. This data also through iCloud, it can go to your physicians, to the doctors. So your family members also can know. They can also understand that in the critical stage they can also get the information. So this kind of very smart technology we can develop also. So this is our main driving force of this technology. Now, just think, and this is the message for the students. Just think that this kind of innovations is possible. And you can think like in the near future, many diseases, this kind of disease spreading, they're coming. So you can develop this kind of technology. You can use this kind of common technology for the diagnosis. Like this is the main driving force. Like we can bring the test method, rapid test method. It will not take much time. Like for testing, if you take the more than 1 day, sometimes, especially the critical stage, like I see you. In this case, just think, a doctor should know the level of some biomarker or something immediately. They cannot wait. Also, some accidental case, we should know the level of marker immediately. We cannot wait for hours. Other than in this case, patient's life can be endangered. So we should. That's why we have to develop some kind of sensor that can give you the information immediately within a few minutes or few seconds. Then doctor can take the immediate actions, kind of surgical action, something, take the action. Then only can save the life in some critical situation. So here is some examples. Like this is the first, I think I am just telling, this is the glucometer. Like they have, you can see the chip, this is the use and through kind of chip. Many of you have seen already. Like you can prick your finger, you can drop the blood, then you can see the digital value of the glucose. Similar kind of device we can develop for other disease, or even for the SARS-CoV-2 that already many devices already came to the market. There's a rapid test kit also available. So this is called actually biosensor device that can help you rapid to take the rapid decisions. The another thing, this is pregnancy test kit. That is also already in the market. There is a color change. If pregnant, then you can see the color. If there is no pregnant, you will not see the color. So this kind of color change information also will guide you in information. Also, as I mentioned, we can print the sensors also because of the advanced printing technology. We can print the sensor also on skin, and it can detect that is from your sweat sample, because our sweat contain many analytes, many disease markers also may contain. So there also we can, this kind of sensor easily with data can be communicated via Bluetooth to your mobile device. It's very simple because of the advanced electronic technology or PCB technology we can use here. We can easily transfer the data from here to your mobile device. I think some different also smartwatch, they also can detect different kind of sensor. This kind of device already in the market. So here at IIT Kharagpur, we are working also with this kind of very advanced light sensors. So we can detect many diseases at the same time and quickly. Where we are using mainly electrochemistry, we have nanotechnology, we are using the biotechnology method, surface functionalization method, and we are designing device, we are patterning the surface, and we are using different nanoparticles mainly. Because nanoparticle has a unique role in this sensor device development. And basic chemistry, because we need the signal amplifications so that we can amplify the signal very low level to high level, so that if you have very low amount of the disease, that we can also detect. So this is the main things, like these presentations basically is very interdisciplinary, because we are not using only the biology, only the chemistry, we are using engineering part, we are using nanotechnology part. So it is kind of the mixtures of our knowledge. So it is very much collaborative also. We are collaborating with different foreign institute also, with all over India also we are collaborating, and then we are making some device. Okay, so that's our main goal, to develop a biosensor that can detect ultra sensitive. This is very important, because ultra sensitive, without this we cannot detect the disease at this stage, right? Here is some pictures I am showing, like smartphone integrated sensor, say C1 electronic part. They are helping actually signal processing, and because you can see this red arrow, this is actually sensor where you drop the sample and all the reagent actually inside this device. Then the signal will go through this electronics part will help for processing the signal, and we will see in the app, mobile phone app. So this is very smart and very user-friendly method we can use. And you can see, like a SIM card, our sensor actually very small, and actually it's miniature, easily you can carry. And you can see there is so many channels, like there is some different lines you can see. Here you can inlet and inlet 1, inlet 2, it means here you can inject the samples, and then there is inlet 2, maybe there will be some chemicals, and now this sample and chemical, they can react, they can mix together, they can come to the sensor surface, and it will generate the signal. That signal you will see in the smartphone. Also, we can amplify the signal by using different chemistry. That is called signal amplifications chemistry that we can use also. So that is called like different kind of enzymatic reactions and redox reactions, and that also we can use. Also, you can see like just you can drop the sample, no need any kind of sample processing. Especially the ELISA method. Some of you know the ELISA method. For that you need lots of sample processing. If you see in the laboratory people how they're handling the blood samples before you're getting your data. Generally you are sending your sample, then morning and evening you are getting the data. But do you know the history behind this data? Many expert persons, they are handling your blood samples. They need the many steps. Like they are processing, they are purifying your blood sample, they are separating all the blood cells, they are getting the plasma or serum. There is many steps. But we can do all the things on a chip. This is a chip, on a chip. Just drop the blood, all the processing can be done inside the chip, then you can see the data. So this is the called the integration technology that we can do. So you can see the whole things. There's the electronic part, there is some sample processing part, there is some chemical reaction part. Everything can be done on a chip. This is very advanced technology. So there, I'm going to show you some sensor basic things, just fundamental knowledge actually. The students you should know first, fundamental knowledge. What's the behind the sensor? What's the reactions going on the chip? Just one example you see here. Just we published this one in a very good journal, the electrochemistry journal. They actually use very basic chemistry. Actually, who are engineer, they maybe don't know, but sometimes they are not interested in the chemistry behind the sensor. But if you know the basic mechanism, it will be easier for you to understand the whole sensor. So I will show you first some few fundamental reactions or some basic chemistry behind this sensor. You see, here we developed a enzyme-free electrochemical biosensor. So maybe you just try to understand here, like we use here platinum nanoparticles, because the platinum nanoparticle helps actually oxidation reactions of hydrogen very fast. This is the electrochemical oxidation, the very fast on the platinum nanoparticles. And with this nanoparticle, we can use some chemical like MB, the methylene blue, and we can use these hydrogen oxidation reactions can happen in the presence of methylene blue also. But these reactions actually we are measuring by measurement of the cyclic voltammetry. This is a electrochemistry measurement. Cyclic voltammetry, actually you can see the y-axis the current and x-axis in the potential. So we are measuring this current with the different potential window. First, this is the oxidation and then reductions. So here you can see the platinum nanoparticle hydrogen, they are reacting very fast. That's why we are getting very high current. Then if you apply the hydrogen and methylene blue, then see the reaction changes, current decreases. So because of the presence of methylene blue actually is hindering the reaction. So you can see here the schematic on the left hand side, on a electrode surface, we can immobilize a primary antibody. It is an antibody that can detect the malaria, PFHR to a malaria marker, disease. This is very generic sensor actually. You just replace the primary antibody with some SARS-CoV-2 antibody, then it can detect the SARS-CoV-2. As I told you, it is very specific. So which antibody you are using, it depends on that you can develop a sensor. So here I am saying it's a malaria-like sensor. So we have to use a malaria-specific antibody. So this malaria antibody will detect the malaria antigen. So malaria antigen will bind here. We can use a secondary antibody where this methylene blue, I told you, methylene blue, platinum, hydrogen, there is some reaction. So we are actually conjugating secondary antibody with methylene blue. Okay, and we are always using the platinum nanoparticle and hydrogen inside the solutions. So you can see here, because of the methylene blue, this current actually decreases. You can see the right hand side, this is a chronokilogram. Chronokilogram means by applying some potential, we are getting some charge. You can see if we increase the methylene blue concentration, the current actually will decrease. From here and here, you can, here charge will decrease. So if you see that the number of malaria marker increase on the sensor surface, then number of the secondary antibody that conjugated with methylene blue will increase. If more methylene blue means this hydrogen concentration on the surface will be less and will get the lower and lower current. So it means if the malaria marker increase in your sample, then more methylene blue will be on the surface and current will decrease proportionally. I will show you some data. As you can see the right hand side, this is a chronokilogram data. And you can see different concentration of malaria marker, like 1 pico, 10 pico, 100 pico. So we increase the concentration of malaria marker. See the charge slowly decrease. And accordingly, we make a calibration plot. So if your malaria marker concentration, increases, then slowly your charge decreases. So from there, actually, we can detect your sample how much marker present. Marker means analyte or target present. That we can detect by using this electrochemistry. See here, actually we try to detect this marker in the saliva samples. Why we try to use the saliva? Because sometimes you have to think about not only the blood sample, because sometimes it's very painful to collect the blood. That's why it is telling the invasive method. So we have things are non-invasive. Non-invasive means that no need to prick your finger, no pain. So kind of non-invasive sample means saliva sample is the one kind of non-invasive sample. Blood, not blood, urine sample, sweat sample. If from there we can detect the disease very easily, that is called a non-invasive diagnosis. So we tried the saliva sample and we check. This is the cyclovoltammogram again. So if you use this potential window, there is almost, they are not showing any kind of interference. You know, our blood samples and saliva sweat, they contain lots of other species, different ions. So during the electrochemical measurement, they also can oxidize, they also can reduce. So they can show the some interference effect. They can increase the background current so that your main signal can decrease, your main signal can be affected. So that also you have to optimize, or you have to check which potential window you have to use for your biosensor. This optimization is very, very necessary. So that we check which potential window should measure. Also, we check the specificity. As I mentioned, no, your real sample contain many other species, so they should not interfere. Also, maybe see other marker, like you are going to detect the PFHRP2. Suppose this is the black one is the control. Control means there is no target, no analyte, no marker. So if it contains other marker, so see you can see this almost control data PFLDA is another marker. They are almost similar data. It means they are not interfering actually. But if there is malaria PFHRP2 present, then you can see the signal change. It means your sensor is very, very specific. So your sensor should be specific. That is very, very important for biosensor development. This is information for my students. So you have to always think about the specificity, and also at the same time sensitivity of the sensors. You can see I mentioned here limit of detection. Limit of detection means how low, how much low you can go with using your sensors. Means maybe you can. I think many of you know the picogram. This at the very, very low level. Even you can go the femtogram. Even your sensor can detect very, very low level of the marker. This is called the ultra sensitive. Means if it is very, very sensitive, it can detect very, very low level analyte. That also you have to care. Here the another example that I was saying of ultra sensitive. Now the question, how you can develop ultra sensitive? Sometimes you have to use some chemical reactions. That I was always saying, no? That you need some chemistry, chemical amplification. So what is this amplification? Amplification means here you have to use some chemical so that they can react each other and they can accelerate the electron transfer process. So you can see here, I just showed that we can use some basic chemical, but that chemical selection is also very, very crucial part. It is not like it's randomly you can select some chemical. You have to check like if they are reacting very fast or reacting very slow. We check the ruthenium, methylene blue, this kind of reagent, they can cyclically react and very fast, and they can accelerate the electron transfer process. So here you can see the cyclic voltammogram sum. When you mix this chemical like TCl, methylene blue, and ruthenium, you can see there is a big change current. Because of this, actually, we can say if we mix this chemical and we can amplify the signal. I mean, see, if there is no target, say something like if there is no target, like no PFHRP2, then secondary antibody that conjugated to the methylene blue that will not bind. So if methylene blue absents, then this reaction stop. The DCF cannot react. Then this DCF cannot react with the ruthenium. Only in the presence of methylene blue it can react. So presence of methylene blue is very important part. So if there is no target, no methylene blue, your reaction will be very slow. But if there is target present, means your analyte, then methylene blue will be there and they will react very cyclically, very, very fast. And because of this cyclic reaction, you can achieve very, very low level detections. That's why it is called ultra sensitive detections. So we can see here, we measure different concentrations of marker and almost femtogram level we can detect even in the blood samples. So that's why we say this is the ultra sensitive. We check the specificity also. You can see the control. When you check, this is almost no change, but when you target present, you can see the big change. Also, at the same time, you have to think about the stability of the sensor. Maybe you develop some sensor, you cannot store for the long time, then how will you commercialize this sensor? Because you have to bring to the market for the common people so that they can use. Also, you cannot store, always you cannot store maybe in the refrigerator. Sometimes the antibody, they are very much stable in the 4 degrees Celsius in the refrigerator, not in the room temperature. Then you have to think how you can stabilize this with using some stabilizer, with some chemical you have to use. Because in the rural area somewhere in the in the summertime, the very, very high temperature in some area of the region in the India, maybe sometimes there is no refrigerator also available. Then how will you use your device? You have to think how you have to develop a sensor by using a common method so that we can avoid the refrigeration system. So we have to check the stability of the sensor in the room temperature, even at the higher temperature. So this is the actually very big challenge for the diagnosis systems, how you can overcome. So that I listed the problems. So this is the message for my students also. You have to think these kind of problems, and then you can find out some research area you can focus on, like how you can develop a highly sensitive sensor, how you can develop a highly stable sensor, how you can amplify the signal. If these kind of questions, if you can in your mind, if you can address those problems, you can develop some new work. Okay. So that's why I just told already that fast detections, bench to bedside detections, ultrasensitive detections is very, very important. So it should be the near patients, not always laboratory based. So we need some new detection technology also, not always the laboratory based. I will show you some detection technology so that you by yourself, it can be handled by yourself. No need to depend on some expert person. That is called washing free some immunosensor. Immunosensor that we are using some antibody antigen interaction. So what is this washing free sensor? Means, as I told you, the expert person inside the lab, how they're processing the blood sample. During the processing the samples, they are immobilizing some chemical, some biochemical on the surface, then they are washing, and many steps they need washing. And for handling these things, we need expert people. But end user, they are not the expert. So what they can do? They can maybe drop a sample on the sensor chip, and they can wait for some time, minutes, and they want to see the value, how much, like for glucose, how much glucose present in the blood, or if there is a malaria or maybe COVID present or not, they wanted to see the value, some digital value. That can be done by using a washing tray. That's why we need a sensor. So this is a sensor chip that already pre-immobilized some antibody that already done inside the laboratory, and this is actually commercialized sensor chip. So what is the role of the end user? They will be provided a test tube. That tube will contain some reagent. Some basic reagent will be there. So they can drop their blood sample. They can drop their blood sample or maybe sweat sample or urine sample inside, and they can properly they can check it and they can drop on the sensor chip. Once they drop, if there is some target present, that is the marker. If present, that will immobilize on the sensor. If not present, then what will happen? The secondary antibody that conjugated with some tag, maybe enzyme, maybe methane blue, just I told, no. So this tag actually will help for signal generation. So if see target present, then this tag will be very close to the surface. And if there is no target, see that this my cursor. So here, no target, then this tag will be very far from the surface. Now we can apply some potential. If potential apply by using some chemical, because this tube already contains some chemical, this chemical will react and it will transfer some electron. That reaction will be very, very fast. In the case of the left hand side, you see if the target present. Why? Because we are very close to the surface. This reaction will be very, very fast. On the right hand side, this scheme showing it will be very slow because it is far from the surface. This reaction rate will be slow, electron transfer rate will be slow. So basically, you are not doing anything. You are just dropping the sample and you are waiting for 5 to 10 minutes. Maybe you have to wait. During this waiting time, this reaction will happen. So if target present, the fast reaction will happen. If target not present, then slow reaction will happen. Accordingly, you will get the result, the signal. So you can significantly simplify the procedures. So you can see here some data that we published in a good journal. So here you can see different concentrations of prostate-specific antigen that is caused for the prostate cancer we detected. Also, this is another washing-free sensor where we detected the troponin I. You can see troponin I is responsible for cardiac disease, the heart attack. That also we can detect by using this washing-free sensor. But here actually we use some different chemical so that we can minimize some interference. As I told you, our real sample like blood or others, they contain so many interference species. So we have to avoid by applying the high potential, the high electrical potential. You have to apply very low, some low, so that you can minimize the background current. So that you can, so low interference washing-free biosensor is important for point of care device development. So what kind of interference can present in our blood sample? It can be like ascorbic acid, right? Because vitamin C, this kind of the actual interference, they can oxidize while you are applying the potential. Or uric acid, our blood sample contains uric acid, acetaminophen. So many other species like glucose also they can oxidize. So you can see the potential window where they are oxidizing. You can see around 0.4, 0.6, 0.6. So they are oxidizing in this region. So you should not apply in this region potential. You have to apply lower around this potential. So that's why we apply here 0.05 volt near here, so that we can avoid this kind of contribution, so that we can minimize the background current. So we applied this scheme and we detected the troponin I, different concentration, troponin I we detected, and the limit of detection is the 10 picogram. So what is the limit of detection? The lowest concentration that you can detect by using the sensor. So now let's come the main challenge that I told you. Like everything you have to bring that's why on a single chip, right? This sample processing. Here I mentioned like sample purification, sample amplification, sample injection, sample detection, sample mixing, separation. So we did so many steps while we are using the blood sample for detection, say marker. You can see here is a chip here. So you can see there is a different inlet. Here you can use some blood samples. Here you can use a buffer samples. Here you can use some chemicals. So now push a button. Then all things will start. So sample purification will start. Sample injections is just injected already. Mixing will be done in the microfluidic by using some microfluidic channel. Also you can heat the samples also on a chip. Sample then detection finally will start and you will get the value on your smartphone. So just see the lab work, what we are doing inside the lab before development this kind of point of care device. What we are doing this kind of steps separately. You can see the workbench. Maybe here one of my PhD scholar working the purification step. Here another scholar may be working on the sample amplification step. Here maybe some mixing step, and they are integrating and he is getting the total actual value. But on chip now, all the lab work is integrated. See, that's why you're all lab on a chip, right? So that's why it is called lab on a chip. All the lab work can be done on a single chip. So already commercial available that I told, that glucometer, meter, pregnancy test kit. So what's the end user doing? Just drop the sample, they're waiting. But you don't know inside, but behind this, behind the getting this value, what's happening inside the this meter? What's happening? These kind of things happening actually. Sample purification happening, amplification happening. Then finally you are getting. So individual, and they are getting just the value, and they can see the actual result. Yes, they have this disease. How much? So this is the very important things for the electrochemical sensor. They can detect how much. But for the colorimetric case, it is difficult sometimes because you can say yes or no. If you have disease, color change. No disease, no color. But how much? It is the advanced level that you cannot understand. But electrochemical signal, you can see the value. Like 108, 107, 50, that value is. So that's why lab on a chip is a very huge market, huge impact recently we have. What is the unique advantage? That's just I told. Like a very small device you can make. That is called miniaturization. Low reagent volume. So sometimes, when you are sending your blood sample, sometimes it's painful, right? They are taking few ml samples. It is very painful sometimes. And it is also some psychologically you are affecting also, right? Oh, that much blood sample they are taking, right? But in lab on a chip impact, just one drop blood, just you can prick the finger, just one drop blood, it can screen, it can detect many diseases. So we need very low volume sample, low volume reagent also. As I mentioned that last slides, that we need some sample amplification, further we need some reagent. Because of the very miniaturized size, we need very low amount of the reagent. So this reagent cost is very high also, very highly expensive reagent we are using. If the sample volume when we are handling very high, then necessarily the cost also can be increased. Sometimes we are paying few thousand rupees for single detection diagnosis, right? But if you can miniaturize the volume, sample volume, very low amount of the reagent if you can use, then naturally your price will go down. Few rupees you can pay. That's why we are saying this is an affordable technique. This lab on a chip technique, that's a very affordable technique. This low reagent volume. Okay. So rapid analysis can be done because all the steps we are not doing one by one. This automatic, simultaneously going on, and within a few minutes, even the second, we can determine all the value. So automations we can apply. And portability. And just I mentioned, because of the miniaturized system, easily we can bring one place to another place, and when patient can keep these things in their home. Okay. Now, so it is an indicative application. Say this is you can use for medical diagnosis. Not only the diagnosis for medical purpose, it can be used for the environmental monitoring also. So in this chip, not only the blood sample, you can use some soil sample. Why? Maybe your soil, where you are using the drinking water, water coming from near the field. So your soil will be contaminated with some pesticide. Maybe some huge amount pesticide, huge amount fertilizers farmers they are applying. That can contaminate the drinking water also, right? So how much contamination there? That also you can detect. Instead of blood sample, just use the soil sample. Use the drinking water sample. So you can determine the level of contamination. Maybe arsenic, right? Level of lead, amount of lead present. That also you can detect. So this is the scope you have. So that's why LOC, lab on a chip, it has the huge market. Lots of funding available, many funding opportunity available you can apply. Now I will show you some currently available, some example technique. Let's start with the glucometer. Okay, so glucometer, it's very cheap method, very cheap, very low price. I mean, you can buy just one time this device, and this chip actually use and throw chip, one time single use. They are not much expensive. But you can see the third image here. You have to pre-colorize your finger. Just think about what doing. We are always injecting insulin, or regularly they're injecting insulin, or I can say the diabetic persons. Just think about before a meal, they always have to check their glucose level. Huh? They have to prick finger. How many times they can prick in a day? And it is very impossible when nowadays actually very younger generations also they are affecting. You can see kind of like babies. Sometimes they are also affecting like small baby. Sometimes they are also diabetic. So how many times you can prick their finger before the meal? So that's why we should develop some smart technology so that they can detect their glucose level from your maybe sweat sample, from your saliva sample. But this kind of device we have, some smartwatch we have. Some company they already bought in the market, but they are highly expensive, very high cost. Sometimes, sometimes we cannot think this one for some common people, some in the developing country, they cannot buy for all users because they're very expensive. But they are non-invasive because in order to prick your finger, right, they can monitor from your sweat sample. But the problem is that they're expensive. So we have to think about how we can develop a new technology so that we can the price of the device can be low. So lab one is PCB. PCB is a printed circuit board. I think you already have seen this kind of printed circuit board in computers or some electronic part. So they are very cheap actually. So this printed circuit board we can use for diagnosis. So this is just a lab on PCB with printed circuit board we can use for diagnosis. And all the lab work can be done. You can see this channel on PCB. So sample processing things, sample heating, everything can be done. And we can immobilize different, sorry, we can fabricate different electrodes. So you can see working electrode, counter electrode, different we can fabricate also. Everything can be done. You can see this is the adapter and connection area. So this is a actual diagnosis whole system can be implemented on a single chip, that is printed circuit board. And we can combine with some microfluidics, we can combine some very small, it is called some needles, but that is not kind of the needles that we are using pricking for the fingers. This is the micro needles that is very, very small size. It will not hurt your nerve systems. It will absorb your interstitial fluid, or you can say like sweat sample. Then this sample will go through your microfluidic channel. It will come to the working area and it will start reaction if present in your analyte. So this kind of work actually we have done at different universities, and we detected like glucose level using this kind of printed circuit board systems. So here you can see one image where we developed the glucose detection on the printed circuit board. Here all the microfluidic, the sample delivery means you can see the tubes. Here you can, like for the lab, this is the lab systems. We can flow the glucose and it can come to the printed circuit board, this sensor surface, where we immobilize actually glucose oxidase on enzyme. So there, if your sample contain glucose, it will react and you will get the value, like this kind of value. See the glucose concentrations and the current. If glucose concentration increase, your current also will increase, like this. So this is kind of some examples, kind of these, we can design. So here actually you can see some very basic study, printed circuit board. Basically printed circuit board while we are fabricating in the industry, so there is lots of contamination. So who will work on the biosensor area, if you want to use this kind of platform, you have to think about how to use this substrate. It is not kind of just directly take the this kind of printed circuit board from your computer, from your broken computer, and let's use this for the sensor. It is not simple. So you have to go for some cleaning step. We have lots of contaminations. Contamination is very, very important factor for your sensor. So you have to clean the surface. If you see, before cleaning, you can see there is almost straight line, no signal. But after the cleaning, you can see the very good signal. So like this, that's you have to think about. If you think about your future to developments of sensor using very common substrate, so cleaning method is very, very important. Also, we use the sensor. You can see this is the printed circuit board. This is the full device. It's very portable. Very, you can see very small size. You can easily integrate with kind of a very smartphone size with some electronic device, and you can integrate with your computer. You can see the data. I mean, with IoT, Bluetooth, you can transfer the data. I mean, it is very easy to develop a device. So we use here some S mutants bacteria detections. You can see I also image like a gold PCB. On that surface, I immobilize one antibody, and then we block the surface. Then once your solution contain the bacteria, it will immobilize on the sensor surface. Accordingly, we can measure the impedance. Here actually we measure the impedance, and based on impedance, actually we measure how much bacteria present, how many, number of the bacteria we can count also. So this is the smart technology, like some wearable. You can develop some wearable sensor, and no need also extra battery also we do it. We can make some self-powered battery. I mean, as I told you, like our solid sample or interstitial fluid, it also contain glucose. So that glucose can react with some chemical. It can generate some energy because any electron flow, So that electron flow from there, we can store the energy. That energy can help to run the electronic part so that it can transfer the data via Bluetooth to your mobile system, or you can send the command from your mobile system to your sensor without any battery also. That's why it is called a self-power. So this is also very important applications of the biosensors that you can use a self-power sensor device. And I use, sometimes you have seen a non-enzymatic method. So why non-enzymatic? Because sometimes we are using like glucose oxide, which is enzyme for glucose detections. But at the very high temperature, it can easily, so they are basically protein, right? They can denature, means it may not work at a very high temperature. So you have to think about how you can avoid the floating, or by using some nanoparticles that can react with the glucose and provide some signal. So we check some copper oxide nanoparticle can react with the glucose specifically to show the signal. So we can avoid the using some enzyme. That's why it is called the non-enzymatic biosensor. So we measure the glucose, fructose, galactose, and other, because they present at in the blood sample. So they are the interfering species. So they should not hamper the detection of the glucose. So here actually I already shown you that this kind of device actually we developed while I was in the Michigan State University of the US. So, and he actually, Professor Lilo is my kind collaborator there. Actually, we are developing this kind of, see the smartphone, we can easily integrate via using just cable connections, and you can integrate and with your sensors, and just see you are getting that value immediately. This is pretty simple. Also similar kind of a washing device, maybe sometimes you need the many chemicals. I told you about signal amplification. That chemical you can use also on the paper also. Like you can see the circle side, they are paper basically. On the paper, you can use many chemicals. You can keep in the dry form, and you can use some special stabilizer reactors. Otherwise, the high temperature they can easily damage your sensor sensitivity. Then just keep on the top of the sensor and drop the blood. Not only blood, I told you can use the urine sample, you can use solid sample. Once you drop the sample, it will dissolve all the chemicals. Then it will come to the sensor surface. Okay. And then this is the kind of the adapter, means you can insert this chip on your like smartphone. Then you can see the value. Like this, very easily we can develop a sensor. This is I'm just teaching you a very generic part. You can use this one for any kind of sensor development. So this is the generic part, like so how we can develop a sensor. Maybe my students should know the basic technology. Maybe your sensor surface should contain a primary antibody. If your sample contain antigen, that is target, that will bind with the antibody, and then you can use some secondary antibody. So see, antigen-antibody interaction, they can block the surface. Now, you see the methane blue ruthenium reaction, just I told you, that reaction can be inhibited because of this kind of barrier. So signal will naturally decrease. See, when you increase the target concentration, slowly the current also decrease, a charge also decrease. This is the actually case of the hindrance effect of the and because of the antigen antibody binding. Also, opposite way also possible. Maybe you can use some tag. Secondary antibody, you can use the tag. So sometimes your tag can help you with signal generation. So if number of antigen increase, number of tag on your sensor cells can increase. That can increase the signal. So both way possible. Okay, now the final slide. Slowly I am coming. Not this is the final. I'm just going to tell you the future prospects to my students. Means there is lots of scope in this area. Biosensor. Like you can develop the portable device that can be wash-free, that can be smartphone integrated, that can be printed, that can be wearable. You can develop a T-shirt. Where in the T-shirt there actually you can print some sensor and that can detect the virus from your sweat sample. Right. So this is a wearable sensor. And I just told you that you cell pod. Even the fuel cell also we can integrate with the biosensor. So we don't need basically power supply from the outside. See, I like always these lines for my students, like breakthrough discoveries cannot change the world if they do not leave the lab. Right? So whatever you're doing, please try to bring at least for the society people. So that's why I always use the bench to bedside. See, this is my common logo, the bench to bedside. Bench means inside the lab whatever you're developing, then bring to the market, so that people they can use, they can be benefited by your innovation. Otherwise, no use by using this kind of technology, right? So people should be benefited. So this is called a translation. You have to translate your research. This is the lots of future scope you have. How you can translate. So this is the translation research you will think about. The translation research means whatever the basic research you are doing, you have to go for the clinical research, you have to go for some trial, then you can bring to the near patient for diagnosis. Without maybe like animal study, maybe you have to go through, then you can bring to the market. I mean, you have to go for investigation if it is useful for the end user applications or not. So you can use different kind of nanotechnology, as I mentioned, different kind of nanoparticle you can use for signal generations. Also drug delivery, you can use different nanomaterial also for drug delivery in some specific area, and it can destroy the like cancer case, it can destroy the cancer cells, and you can use for some therapeutic application. And translation research means currently this kind of work already done for the COVID. I already told you the general concept. Kind of COVID sensor also, some sensor rapid test kit also available in the market. So you have to think about in the future also many new diseases also come. How this NanoBio sensor technology you can use for the diagnosis approach, and which point you should address, you should consider, right? So kind of things like it should be easily accessible, it should be affordable, miniaturized, so they can use. At the same time, it should be very specific, so that other parameter present inside the samples they will not hamper, and they should be very, very sensitive, or you can say ultra sensitive. This is the last message for you. And the commercializations of the device that I told, you can integrate all the technology. That is the very integration technology is very, very important, because this is very much multidisciplinary technology. And here sensor, here electronic, here app development. So you can see here like the fabrication technology, lots of things involved. So like this way, you can develop a very simple chip, very small chip, and you can easily integrate with a smartphone kind of things. You can think about it. So thank you, all of you. Thank you very much, and I want to acknowledge all the collaborators, my national collaborators, international collaborators, and I want to thank my institute, IIT Kharagpur, who is dedicated to the service of the research. So thanks a lot. Thank you very much.
So it has been indeed a pleasure listening to you and watching the slides which you prepared for all of us. And we hope in future we'll be having you on our portal in offline, like it will be a classroom, situation, sir. We want our young minds, to listen to you in offline mode. We will be looking forward to have you, on our campus.
Thank you very
Now,
much.
welcome, sir. It's our pleasure. Now we'll have Dr. Shankar Shankar, sir. Morning. Sir, a few questions have, like, we are expecting a few questions for you. So, would you allow
Yes, yes.
us to we'll pass those questions to you?
Yes, sure, sure.
Sir, one of our students has asked this question: does nanotechnology can detect early diseases?
Yes, that's I taught you that nanoparticles they can be useful for the early diagnosis. But how, right? Okay, I'll show you. That's why I mentioned some. Let's show you some slides, right? So this is genetic technology I taught you. You can modify this technology. Let's come to the sensor. Here, you see this image. Here the secondary antibody is conjugated with MB, the methylene blue, right? Now, instead of methylene blue, you can conjugate with some gold nanoparticles. Okay, this is as I am saying, general topic, tag. So you can use some nanoparticle tag. That nanoparticle can specifically react with some chemicals. Okay, now this because of and if you know the nanoparticles, they are very much reactive. Some that technology, that reactions actually you have to, that's why I told you have lots of scope. Maybe some platinum nanoparticle, or maybe some gold nanoparticle, even some core cell type nanoparticle. So alloy type nanoparticle, different nanoparticle you can use. And those can help you these reactions very fast. Not only the fast reaction, because nanoparticle they are very stable at the high temperature. Right? So you can avoid the stability issue also. This is one thing. Another thing, you can immobilize the nanoparticle, you can fabricate your sensor surface also with nanoparticles. Suppose you can drop some nanoparticle on the ITO surface. This is the sensor surface. Because nanoparticle, their surface area, the surface area is very high. The small nanoparticles, because of the very high surface area, lots of primary antibody. Suppose there is 2 nano antibodies, but if you use nanoparticle, you will get lots of antibodies on the surface. So naturally, because your efficiency of the sensor will increase. This is another point by using the nanoparticles. Or this is 2 example. Or another example, sometimes you can use some nanoparticle here that can increase the conductivity of the sensor surface. So electron transfer rate will be highly enhanced. That is another application of the nanoparticle. Like this way, nanotechnology can help you for sensor development.
Thank you, sir. Thanks a lot for taking the question. Um, now, do you have any? I don't think we have any other question. So we are at the end of this session, and we have vote of thanks now. We will request our respected HOD of Mining Engineering Department to deliver vote of thanks.
Honorable Dais and audience, very good afternoon to you all. I feel honored to propose this vote of thanks on behalf of Asansol Institute of Engineering and Management Polytechnic and my own behalf. I wholeheartedly thank you, our guest speaker, for his valuable knowledge which he shared with all our students and audiences. I thank all who has given their all efforts to bring this event a success. once again I thank you one and all and take your leave with grateful hearts. Thank you, sir. Thank you.
Thank you very much. Thank you.
Thank you,
Thank
sir.
you. Thank you. May I leave now?
Yes, sir.
Thank you. Thank you, sir.
Thank you.